So hi, good morning. Thank you for coming today. So it's my pleasure to introduce Dr. Budowle, and it's a double pleasure because it's the first time that the Beto Chair lecture we invite a forensic scientist. So double pleasure. Thank you, Bruce, for coming. So Dr. Budowle, he's a professor and executive director of the Institute of Applied Genetics at UNT Health Science Center. So he received his PhD in genetics in 1979 from Virginia Polytechnic Institute and State University. He did his postdoctoral research at the University of Alabama at Birmingham, studying genetic risk factors for insulin-dependent diabetes mellitus, melanoma, and acute lymphocytic leukemia. But then he moved to the FBI during 26 years, from 1983 to 2009. Dr. Budowle, he worked at the FBI Laboratory Division, and he carried out research, development, validation of methods of forensic biological analysis. So I need to say that Dr. Budowle is one of the founders of the forensic DNA analysis today. So he worked on laying some of the foundations for the current statistical analysis in forensic DNA, and he defined parameters of relevant population groups. So he published more than 490 peer-reviewed articles, and he testified in more than 250 criminal cases involving forensic DNA analysis. So he was involved also in quality assurance standards for the forensic DNA field. He was a member of Sweden, the Scientific Working Group on DNA Methods, chair of the DNA Commission of the International Society of Forensic Genetics. He was a member of the DNA Advisory Board. And actually, very, very important, he was one of the founders and architects of CODIS, so the National Database of DNA Convicted Offenders and Unsolved Cases. Besides that, he was actively involved in the identification of the World Trade Center victim disaster. So during the last year, he was involved and focused on counter-terrorism, specifically on microbial forensics and bioterrorism. He personally analyzed the case of the anthrax letters. Basically, they had to create a new field that is microbial forensics in the FBI. So he's also founder of the microbial forensics field. He was chair of the Scientific Working Group on Microbial Genetics and Forensics. And actually, he serves on different agencies working in working groups related to microbial forensics. So he published many articles and books on microbial forensics, population genetics, deep sequencing. And actually, I think that he was in the editorial board of all top-tier forensic science journals, including Legal Medicine from Japan, International Journal of Legal Medicine, Forensic Science International Genetics, Biotechniques, and actually he's the co-editor of Biomed Central Investigative Genetics. So he received, of course, several awards from the American Academy of Forensic Science, the United States Attorney General, and from the FBI Director's Award. So I'm done. Okay, so he's actually working, he's still working on research, he works on research, and his focus is on the areas of human forensic identification, microbial forensics, and emerging infectious disease. So please, ladies and gentlemen, give a warm welcome to Dr. Bruce Budowle.
Well, thank you, David. In the 3 minutes left that I have to talk now, I'll just say that. So I really appreciate the invitation, and it's great to be here, and really impressive criminal justice program here. And just the facilities alone are outstanding. But I was asked today to talk about bioterrorism, microbial forensics, and presumably you're all here because you want to be budding bioterrorists. Or, that was supposed to be funny, you're supposed to laugh at that. And if I have to tell you when to laugh, the rest of the lecture will be kind of unexciting. But what I thought I'd do is try to give you an impression of what it used to be like and what has changed since probably just right after 9/11, when the first well-documented bioterrorism event occurred in which we had Bacillus anthracis, what you call anthrax, found in letters that were mailed out to various people in the media and in the senators and such. So we have to start thinking first about forensic science and what it is, because forensic science is the application of science in legal matters and legal matters, and we use science to somewhat be more objective in the way we analyze evidence. So when a person sees someone running from a crime scene, it's a chaotic environment and it's a little difficult to recollect or recall what the person actually looked like. And so eyewitness accounts, we know, are not that accurate. So we hope that science and the technologies around will actually serve as better witnesses than what we can do with our own eyes. We have to remember is it's not like it is on CSI. So how many of you are in the program because you watch CSI? Oh, come on, tell the truth. It's about 90% of you that. We got 3 honest people in the back, okay. You watch the TV show, they find a button, and in 44 minutes, because of commercials, you can identify who that person is, what their name is, where they live, how much money is their bank account, and whether they like chocolate or vanilla ice cream. I don't know. But that's not how science actually works. Science is just a piece of a puzzle. And it doesn't have to be beyond reasonable doubt, like you think of when we're in a trial where the government's trying to convict the suspect and the defense is trying to exculpate or defend their client. It's a piece of a puzzle, and it doesn't have to be 1 in a billion chance of ever observing this again. It could be 1 in 10, it could be 1 in 5, but it just adds to the overall picture or puzzle that convinces someone that the issue at hand is beyond reasonable doubt. So don't think of the science as being absolute, but part of what we do. Now, the more powerful it is, the better it is in determining who may have committed the crime, or it's also as powerful in who may not have committed a crime. Now, I keep saying committed crime, but when we think of biological evidence or DNA or fingerprints or whatever, it's not about committing a crime, it's about associating someone with an object or material. It's the rest of the picture around that that goes beyond the science that determines whether or not a crime has been committed. And when we do this, we have to try to think of this in a big picture. Now, as scientists coming in, learning either the social science or the physical sciences, you think about your work as very important. But it leads to a bigger picture. And the first one is we have to determine what the risk is so that we can set up where the threats and vulnerabilities are, and then do all the things about protecting and pursuing. So if it wasn't forensic science and we thought about Katrina, the hurricane that happened a few years back, so we know what the risk is. The risk is people are going to get flooded. What's the threat? Hurricanes, vulnerability. People shouldn't live below sea level, because if you live below sea level, you have things. So what could you do? You could build the houses up higher, move them away, put up a dam, get FEMA in place, all the things we can do. So every one of these things, whether it's a crime, whether it's bioterrorism, whatever, we want to assess these in these ways because then we know where to put our efforts and how our responses will be. Now, when you think of bioterrorism or any event, we don't do it because we expect it to happen all the time. We're actually doing it like we're buying car insurance. You don't buy insurance to plan to have an accident. You buy insurance because you plan not to have an accident, but just in case. We don't prepare for catastrophic events because we expect them to happen, but the effects can be devastating. So this is a picture of the outbreak of foot and mouth disease in the United Kingdom that was a natural event. Mother Nature's the best known bioterrorist. She makes better weapons than anybody else so far. And so we have a lot of experience we can gain by seeing what happens in natural events. This was a badly managed outbreak that cost the United Kingdom over $40 billion in trade, tourism, and other economy. So one event can have a devastating effect. So the preparation is very important, even though it doesn't happen very often. Now, we used to think of bioterrorism and an attack coming from not Mother Nature, but from man, is the idea would be that this Russian submarine, I guess we can say Russians today are bad, not so politically correct, since some Ukraine event I don't understand. They would come along the harbor in New York, spew out a cloud of some microbes, and it would infect the populace, and everybody would get sick, and some would die, and it would be an effective weapon. Now, it wasn't believed to be a very effective weapon because it's not easy to disseminate microbes in the environment. They're very fragile. They die. You have to have perfect conditions and set up to make it happen. So most people really didn't take it seriously what happened. So I like to look at history, and most of you in this room won't even know who these two guys are, right? Do you know who they are? Good. These are actually one of the hottest comedy teams in the U.S. back in the 50s. This is Dean Martin and Jerry Lewis. And I found this Life magazine not because of Dean Martin and Jerry Lewis, but up in the right-hand corner, what to do about germ warfare. So this was the philosophy of the 1950s. And they had invented something really cool that was going to help them in our biodefense. It was called filter paper. Okay? And the best way to defend was to be able to collect something on that filter paper and get a rapid detection. Because if you can detect quickly, then we could respond to what the threat may be. And without that, the U.S. was going to be really woefully inadequate in its protection from these kind of attacks. So you think about it, filter paper. I can tell you in today's world, that's exactly what we're still thinking about, filter paper and collecting things. So 60 years later, something like this, where you have scientists sitting up with some device that looks like a nuclear bomb right after World War II, during Korean War, and no one's paying attention to them up on this roof. And you notice there's a couple of things about them. One is open-toed high-heel shoes. Whoever works in a lab, not going to happen. And they're not wearing gloves, so you know it's a little bit of a different time frame. And the idea was that they would put these samplers into this device, and as the air passed through, it would collect any particulate matter, microbes or whatever, and then you could analyze them subsequently in the laboratory. And then you would take the filter, and put it into a Buckner, the Buckner thing, and mouth pipette. So I guess if you're mouth pipetting microbes, you don't have to worry about wearing gloves and that. So you can see him not wearing his gloves. And then take it, add on some nutrient, and then put it into your biosafety level 3 incubator pocket and let it grow, okay? So this was the idea. So then we'd be able to detect the microbes here and then be able to respond. Obviously, I think we've gotten a little better since then, but we didn't detect ahead of time this event, and these are just 5 of the number of individuals who got exposed to Bacillus anthracis, anthrax spores, in the attacks that occurred right after 9/11. So remember, the country had just been attacked, planes had crashed in the World Trade Center, the Pentagon, and one crashed in Pennsylvania, and within a few days after that, the second shoe drops, and then we have these attacks. But this was not recognized at first. In fact, the first person, Bob Stevens, who was the first identified person who died from the attack, only 5 died. These are not all of them that died, just a couple of those up there died. The others were exposed. And we had to start responding to this event. So the government rallied its forces, spent billions of dollars, and many of you here may ask, well, why should we spend so much money on something? As I told you, was the only event that had occurred, because it seems like it's out of pace. We've only had 1 attack, correct? Well, of course, I didn't tell you the truth, otherwise the story wouldn't be any good, is that mankind has been actually perpetrating biowarfare from as back into history as one can imagine. Even before the Romans knew about it, the Assyrians, 600 years before the Romans, found out, and they didn't know exact cause, but they knew that if you took dead, decaying bodies and put it into the water supply, it would poison the water. So they would use this as biowarfare against their enemies in routine war efforts. In fact, up until the 20th century, this was a routine approach used to battle your enemies. The 14th century, up in Ukraine, the city of Kaffa was under siege. And the Tartars soldiers had come down with plague. And this was right around the beginning of that 3rd pandemic in Europe. It killed about maybe a third of all Europeans at the time. And so they had this problem now because their soldiers were sick, they were dying, so they decided to turn that disadvantage to their advantage, and they catapulted their dead soldiers over the walls into the city to infect the inhabitants. So it was another form of biowarfare. When the British and Spanish came to the Americas, they met what's called a naive population, a population, Native Americans, who had not been exposed to smallpox or measles. And they gave them infested clothing, blankets and things to expose them to this, and more Native Americans died from disease than they did from any other form of traditional warfare. So they knew what they were doing. There was good documentation of that. I think one of the most interesting ones was the Germans. They had a clandestine lab in Maryland that was growing what's known as glanders. Glanders is a disease that attacks horses and mules. It's a bacterium called Burkholderia mallei. Remember that for the test at the end, because that's going to be the hard part. And they were culturing it and then going down to the stockyards and infecting the horses and mules with glanders. The idea would be that they would get infected, the horses and mules would go over to Europe, and then they would infect the other animals there, and it would help turn the tide in favor of the Germans. So that's a pretty bold way of moving forward. And most of you might say, so what about horses? But back in World War I and prior to World War I, the cavalry was such an important part of our forces as opposed to today. Now, the Japanese probably had some of the most impressive biowarfare programs in that, and they actually released plague-infested fleas and dropped them over Manchuria, China, during World War II to again infect the populace. Japanese also had an undeclared war with Russia that predated World War II, so they had this idea that they would then, infect Russian soldiers. They would then get infected and then take them back to motherland Russia and infect the population and turn the war against them. But just having a bioweapon doesn't mean that you can always affect it. And the one thing the Japanese didn't understand is the way that Russians think about their soldiers versus the way we would think about our soldiers. Is when a Russian soldier gets sick, they just leave them on this roadside. They don't take them back. So basically, as they got sick, they just left them there. So it wasn't an effective measure, even though people could get ill. There was a cult that was a pretty sort of out there cult for India, and they came to the U.S. and settled in Budowle in Oregon. And there was going to be an election that was going to affect some property rights and owning that was going to impact on them. So they wanted to turn the tide, so they took salmonella and salted it on salad bars. And that you're going to learn very quickly, do not eat open food, okay, especially in break rooms or whatever, all you students especially, that's all you do. And what they did was about 751 people came down with food poisoning. They still lost the election though, so they didn't work as well as they hoped for. But no one knew that this was a bioterrorist attack. It was only till afterward one of the cult members had confessed years later. So we were not thinking of bioterrorism, we were thinking of food poisoning. Muffins in the break room in Dallas, Texas. There was a nurse, and I'll show you a picture of her later. There was a nurse that took Shigella and put it in the muffins and left it in the break room. And what do people do when they see baked goods? You take it, right? And you eat it. And they were able to do that. They came down with dysentery. The stool specimens were typed, and they matched the strain in the hospital where the nurse worked. And also some people took the muffins home with them, and they were able to get that. So it was another one of these things. This is an interesting one because the nurse had a boyfriend, and he kept coming down with bacteremia. So, they go to the hospital, they treat him. Turns out she was giving him vitamin B12 shots. So the second thing is, besides not eating food that's open, and you don't know where it came from, if you have a lover, a friend, whatever, and they want to give you a B12 shot, don't take it in that, okay? So the last one I want to talk about is the Shinrikyo cult. And some of you may remember in the mid-90s they released sarin gas, or nerve gas in the subway in Tokyo. And this was a pretty aggressive group for Japan. And this was their leader, Ashoka Asahara. So he was a pretty dynamic cult kind of figure. They would follow him, do whatever he wanted. And they had some pretty aggressive programs in that they were trying to get Ebola, which is obviously very much in the news these days. They were trying to aerosolize anthrax and botulinum toxin and use it as a weapon in Tokyo. They experimented with Botox, Botox, the stuff that people do to get rid of the lines on their forehead. It's actually just a very, very lethal toxin. Q fever, cholera, all these things to attempt to attack the populace in there. So this is an apartment building that the arm owned. And just a typical thing in downtown Tokyo. And the inhabitants, or residents, called the police and complained because there was this strange odor coming from on the roof. And they thought that the arm was boiling humans on the roof. So they called the police, and the police came to the building. They weren't allowed in. So a little bit difference between Texas and Japan. If you're boiling humans on the roof, the police here would definitely be in the building. And so they were told to stop doing what they did, and they noticed that there was some gelatinous oozing material coming down the side of the building. So they collected 4 tubes of it and tested them for human proteins, and they were negative. So the 3 that they tested, they threw out, and the 4th one just went on a shelf for several years. Well, after the the sarin gas attack, one of the cult members said, well, what about the anthrax attack? And of course, we said, what do you mean anthrax attack? Well, if you look at this building, you see the smokestack? That's not a smokestack. That is a collar hiding a sprayer. And that is a sprayer that's actually sending Bacillus anthracis onto Tokyo proper. And yet nobody became ill, but they actually were sending it out there. And so we went back to that 4th tube and cultured it, because anthrax is very stable. And this is what the culture looks like. So those colonies are characteristics of Bacillus anthracis. And it was typed genetically and confirmed, but it turned out to be the Stern strain. So if you have a species of bacteria, you can have what are subspecies called strains, and each of them have different characteristics. Now the thing about the Stern strain is it's a vaccine strain, so it's used to immunize animals or whatever. So therefore you don't get ill from being exposed to Stern strain. So now you have to have theories of what would happen. Well, one could be, maybe they didn't know the difference between a vaccine strain and a lethal strain, which was probably good. Second one could be it was a trial run and they were just getting ready for a real event. Or it could be, sort of bad leadership. When a dean or a chair of a department tells you to do something, you do it no matter what. I'm only kidding about that. But the same thing here, could have been saying, you will do this, even though they weren't ready, and could have led on, or a combination of all 3 of these, and no one knows for sure. Now, in the anthrax attack in the US, we were posed with quite different set of problems. When it became apparent that this covert attack, because no one knew it occurred, became an overt attack when the letters were found, how do you find all those letters in the US mail system? An almost impossible task. So we had to collect all the mail in an area, and these are 55-gallon drums filled with the post for that time frame, built into a makeshift biosafety level 3 warehouse. And you can see people have to wear their protective equipment. It's not an easy thing. You can overheat, you have to work in this. How are you going to go through every one of those letters? So you had to actually devise a system where we took basically a garbage bag, filled it up with air that would suck some of the air out, and then test to see if it was a high count in there or a low count. Had a high count, potential letter, low count, probably just cross-contamination. And that's how the other letters that were found that were never delivered. Now, why would a letter cause so much havoc? Because, when you open it, you can see you open it up, you got this powder, and it says you're going to die or whatever is on the letter. So you're forewarned. But this is what happened. You see here. It says this is a simulation of the letters with powder of the same dimensions being sent through the postal system. When mail goes through, it's sorted, but it's it's vigorously agitated. So And so you can see all that powder is rising up, and as that powder rises up, people got exposed. So people working in the areas, or people got letters, or those distributing the mail got exposed, and it didn't take a lot of particles to contaminate somebody and cause them to die. So this was a very serious thing. And that changed the way we think, because remember I said at the beginning, it was very difficult to deliver microbes and as an effective weapon. Now all of a sudden, you can just send it through the mail in a very simple fashion. People get exposed and you can disrupt very quickly. So we had to develop the field of microbial forensics as another arm in the investigation using science to move forward to try to determine who are the perpetrators of such crimes. And it's no different than any other forensic discipline about collecting evidence, trying to extract information and interpret it. But it takes just a different focus. It's on bioterrorism, biocrime, or inadvertent release, whatever it may be. And I'm not always sure where bioterrorism starts and biocrime ends or this. So it's really a collective of using pathogens or toxins as a weapon. So what we want to be able to do is, or wanted to do and still want to do, is extract as much information as possible from biological evidence, this time being microbial evidence, to be able to figure out what happens, and at the same time, not compromise any of the traditional methods. So, because those also are as helpful in trying to determine who may or may not have been perpetrators of the crime. But microbial forensics is a very complicated discipline because it's made up of a lot of sub-disciplines. It can be obviously forensic science, it can be genomics, it could be analytical chemistry, plant pathology, veterinary science, bioinformatics, computational analyses. All these things play in. So it's not as simple as saying I'm going to just be a fingerprint expert and just look at the patterns on fingerprints now. We have to pull in a lot of different disciplines. So it's a very demanding process. And it's very complex. And it's not just as simple as you think, because there's a lot of unknown in the work in this area, and we'll get more into that. But our goal is, as I said, in anything else, is to try to figure out what is the source of the sample. Now that could be either what it is, where it came from, who made it, what laboratory it could be found in, or whatever, with the whole purpose of trying to determine evidence that can be used either for investigations, to further your focus, or again, to help in determining guilt or innocence in a situation. So we can think of all sorts of things. I said, you might want to know what it is. You want to know, was it engineered? Could have been manipulated, suggesting a more sophisticated laboratory than sort of something done in someone's garage. Where it was made, when it was made, to help in the investigative leads the focus, and we'll give some examples later. As a geneticist or a DNA person, I want to think about the kinds of questions that would help in that, and I'm not going to go through all of these, but you're looking for differences and similarities to be able to determine whether or not this came from the same tube, lab, or whatever in the process. And there are many of these kinds of questions came from. You may want to know, does this microbe that I found in this case exist in the environment where the crime was committed? Because maybe it won't be a crime then. Maybe it was something natural. We have to be able to distinguish them. So we have to deal with this in a way that's much greater problem than we ever dealt with when we thought about human DNA. Human DNA, we have one species, set of markers that covers all of you here and everybody in the world. So we can develop a set of tests and it will apply to everyone. With microbes, it's a little bit different. There are over 1,000 known agents that affect humans. Now, if we just look at the bacteria, about half of them are bacteria. And of course, some of them, you get the cold, you get a flu, it's not the same thing, but a lot of them are very serious. So we don't have one target anymore. We have a lot of targets. We need a lot of different tests. And this is a big challenge because the resource and effort is far more demanding and almost impossible to support if we have to design a test that works for every single possible bacteria out there as an individual one. Now, if we add in from the plant and animal side, there are over 50,000 known pathogens that attack plants. So if you thought there was 500 for humans, now if we add in the animals, a few hundred more, and then we add in 50,000 plants, at least you have a lot of work to do for you, so you have a career for a long time. But it's very challenging. So we published a paper on, I'll call this the tree of death. So this is the bacteria that are recognized as potential high priority pathogens, A, B, and C, from the CDC and some of the USDA ones. So if you wanted to start off, these are the ones that we worry about the most. Okay, so we at least know where we're going for, but still, that's a lot of targets and a lot of tests that have to be developed. I already mentioned the overt and covert attack. Sometimes someone puts out a box and writes on it anthrax. Now, I always found that to be the most helpful terrorist, to tell you exactly what's in the box. So they test for anthrax, because that shows you how well law enforcement thinks about things. Well, they told us it was anthrax, so it must be. And that's not a very good way to do things, but that's what happens. But that's an overt attack. You know something's there. Yeah. Whether it's real or not, something you have to determine after that. Covert is something that happens and no one has announced it and it's just waiting to play out. So if you think of the anthrax attack, when the first person became ill, no one even knew this was an attack, just someone became ill. It was covert. And until they found letters that were delivered to Brokaw and to Daschle and others like that, then it became an overt attack. So how do you determine a covert attack from a natural event? Well, we use epidemiology. In fact, epidemiologists are forensic scientists, and all forensic microbiologists or microbial forensic scientists use epidemiology as part of the investigation. So it's not a new science, even though we said, maybe 15 years ago we invented this field. It's been around for much longer. And what you do is you ask questions. For instance, if there was a smallpox outbreak today, would anybody think of it as a natural event? No, because smallpox was eradicated back in the 70s, and there's supposedly only 2 laboratories in the world that have supplies of smallpox, and that's CDC in the United States and Vector in Russia. It's not supposed to be anywhere else. And if any of you just recently heard on the news not too long ago, they found vials of it up in Washington, D.C. area, Maryland, in the lab. It's just been there and forgot to be taken out when all this stuff was supposed to be purged. So you can see it can always still be a threat. We don't know who else has them. If you have like the muffins in the break room and you find Shigella in baked muffins, it's an atypical place to find that in cooked products like that. So that would be another surprise. Or so if there's an outbreak in the wrong season of the year and so forth down the line, so people will look for those. If animals become ill, and then people become ill, you start to wonder what's going on in that, because we don't typically eat their feed, so maybe there's something else in common and so forth. And so all these are used to try to tip the balance. Never absolutely 100% in any one case until you look at other evidence, but you start to weigh whether or not we should be concerned as an event or just respond in a typical public health manner. When I mentioned the salad bar, food poisoning is one of the versus an attack with food. It's one of the most difficult to discern because food poisoning happens all the time. Some of you probably had food poisoning. Some of you had friends or family have been food poisoned. And there's hundreds of thousands of hospitalizations every year due to food poisoning. So how do you determine what's real amongst that background of normal noise in the system? And that's another challenge for us. Now, when someone plans an attack, they have to have an agent that they're going to use, some system that they're going to use to take it from their lab and expose the intended target, and then the environment and the target itself. So when you think about investigations, we have to think about all 3 of these aspects and not just, I'm going to identify the agent. So if you look at a farm, very open area, not a lot of protection, and if you saw a vehicle with a sprayer going alongside the farm, what would you think is happening? Pesticides, something to protect the farm, that would be your first choice. You wouldn't think twice about it. So an individual who wanted to perpetrate an attack on the crops with one of those 50,000 pathogens could put it in a sprayer and go right along the road and nobody would pay attention at all. And that, so this is another thing you have to think about, is what are the delivery devices and what may happen. You can also think of, say, the police, they go in, they break into a clandestine drug lab. They're looking for drugs. They're not looking for other kinds of things. So what are other telltale signs that people may be doing things? You see an egg incubator in a room where they're making drugs, you know they're not making breakfast, okay? But they're not making drugs. And that, so what could it be? Well, eggs are used to culture virus. And so these were things there. So we have to start educating our first responders on what are telltale signs of other events that they may not be prepared to understand. Lyophilizers. If a terrorist was actually concerned about the people who work for them, maybe a shower and eye wash in the area could be another hint. A lot of little things that we take for granted, but the police may not understand it, the fire department may not understand it, or other hazmats in there as they go down line. To make it work, you have to collect evidence. And if you don't collect evidence, you can't analyze it. If you don't collect it well, you may not be able to analyze it because it degraded away. So one has to think about the proper methods to collect evidence, and of course I'm not going to go into too much on that, but there are different kinds of methods that you can do. So consider that there was another agricultural attack on some wheat, or let's say corn, out in Kansas. And so you don't know exactly what happened, so you go to the field and you want to do a sort of statistically set up analysis where every 3 meters you'll collect a corn plant. So that's a good idea. So you walk there, you walk 9 feet, you pick up something, you walk another 9 feet, you pick one up, and then later on you go and analyze it. But as you walk along, there's a plant that's got all the disease on it standing right over here, but it's not on your every 3 meter focal point. Should you collect that as well? Well, certainly you should. So you want to make sure you're just not doing things with only one logic in mind. But sometimes you want to look at risk-based, sometimes you want to be targeted. When the individual Bob Stevens became ill and they figured out that he had anthrax, unlikely in Florida, North Carolina area, they went to where he worked in Florida, and they went right to the mailroom, to the keyboard on his computer, started sampling those as likely sources for that, and got very hot hits and exposure. And other things is you don't know what it is, or if you want to go into a lab and you don't want them to know something, you just want to do a quick swipe like this, it's not ideal. So it's very different. Each one of these have different risks of them and different ways of collecting, but you have to think about them for which is better. Sometimes logical and systematic is good, sometimes a targeted approach is better, and so each of these have to be thought out. But one thing to say as a forensic scientist is a lot of people are trying to learn things to follow protocols, and they want a recipe. That's a bad forensic scientist. What you want to be able to do is have a protocol, but think about what you do and start asking questions, because there are very good situations, even in forensic DNA typing today, where if you follow a protocol, you'll get an answer and it'll be wrong because you didn't think through what the problems were. So this starts to make us think about what we do so that we collect the right things. And simple thinking could be just how you collect evidence. If you're a microbiologist, you want to collect bacteria. If you're a classic forensic scientist, you want to collect fingerprints. So if someone in here, you touched that, right? Yeah. So if someone like you touched that, and I want to collect the evidence, I'm going to swab up all this and collect my bacteria, because that's what I'm interested in. But what I may have just done is destroyed the evidence that would identify him as having contact with that. So we have to start thinking about how we collect evidence, triaging, and making better decisions than just what we would do in more classical situations. So as I said, we had a number of people who died, got infected, and created investigation. There was evidence collected. There were letters found that tried to mislead people into thinking who the perpetrator was. So you may read this and think that this was some sort of attack following 9/11, tied to 9/11. All the evidence does not point to that. So you have to be careful about how you take evidence and understand what people may do. This is the evidence that went to the New York Post, and you notice quite granular material went to that. The one that was in Senator Leahy's letter was very fine powder. So right away that tells us there was more than one preparation. Without doing any more science, we knew that whoever did this made more than one batch. Okay? And then typed them from a non-validated method. So remember, forensic science is all about validating methods. We use the non-validated method because you can't say, I need 2 years to validate a method to analyze this evidence when someone is attacking you at the moment. That's not reasonable. That's not good science. That's not good protection of the population, the community. And this was a good method. It wasn't that. It just wasn't gone through that whole thing. And this genetic profile told us that this was the Ames strain, that one of those species of Bacillus anthracis. Pretty lethal strain. And the Ames strain this is what's called a genetic tree. These are all the different known strains, and the closer they are together means they're more closely related. The further away they are, the more distant they are, but they're all strains of the bacterium. Ames strain, you can see where the arrow is in the tree. Western North America, that's the most common strains. So Ames strain is not very common in the US. So what that tells you is maybe whoever did it didn't go out into the woods or in the fields and dig up the soil to look for a strain and use it. This may have come from another source. So even though it's not a powerful indicator of who did it, it tells us something about who may not have done it. And if it's not coming from the environment, where does it come from? It comes from a laboratory. And the Ames strain was found not in Ames, Iowa. It actually comes from not too far from here in Texas, okay? So now all of you are potential suspects because it's kind of, right? It's close to your area. Most people didn't know that it comes from Texas, but when it was found and found that it was pretty virulent strain, it was sent to Ames, Iowa for further analyses, hence the name Ames strain, and then was disseminated to lots of government labs for research purposes. So it was the government labs and their collaborators who had access to the Ames strain. So, as I said, it helped us another thing. There were people criticizing the FBI for focusing on laboratories and not on the environment. Now, if you have this particular strain, it doesn't make sense to focus on people going out in the environment. So it also helps point things, and you can see how people who are critical don't always evaluate all the evidence to support that, and it does suggest that a laboratory was the right source. Then we would use genetics, DNA testing, to try to reduce the potential candidate down to which laboratories could have been the source and which laboratories wouldn't be. Because if you're just a scientist working on it, as you normally do, you don't want to become a suspect. But at the beginning, everybody who had Ames in their lab was a suspect in that, so you can imagine. And when they did the genetic typing, we'll go more into that, sequenced the entire bacterial genome back in 2002. It was the one in the original collected sample and the one that was found in the victims was identical. So there was no way to tell any of them apart. So it wasn't the tool that was going to be able to allow us to separate out the attacked weapon that was developed versus what was the original material. So after all that hype for DNA typing and all that, didn't get us anywhere. But one of the microbiologists noticed that as you culture this particular sample, late forming colonies had different morphology, different shapes than the main type that was the same genetically. And these, what we called morphs for morphological variants, had genetic variants in them that caused that morphology change. So those were sequenced to find that one little site out of their entire DNA chromosome to identify what was causing that so that we could then develop tests for identifying those variants to screen the evidence. Now, sequencing is really the way to go. Sequence the entire genome. But back in 2002 is very different than it is today in 2014 in a very short time frame, if you think about it. Sequencing is great because when you do analyses and you say, I'm looking for this organism or this target of the gene, you're looking just for that and you're ignoring everything else. So that's called a biased analysis. Not biased because you're bad people or you're thinking evil or that. It's just that you're focusing on one thing and ignoring everything else. Sequencing says I'm going to look at all of it So there's no bias in what you're looking for. So you have a better chance to identify those particular markers inside the bacteria's genome, its DNA, to be able to say that this one is different than that, and this one is similar to this, and so forth. And so at that time, it was done by shotgun sequencing. Same technology was used to do the Human Genome Project that took over a decade and several $billions to sequence one human genome. So you can imagine, of course, the human genome is 6 billion nucleotides long, and bacterial genome is, say, 5 million. So it's 1,000 orders of magnitude greater challenge, but still not something to be done. And it was done by the Institute of Genome Research, Tiger, out of Maryland, to identify this, and it took 3 months for them to sequence that one genome. I can do that in my lab in a day and a half or 2 days now. So you can see how technology has changed. And then you got what are known as polymorphisms, differences that they found between what they sequenced in the original sample. Now the problem with sequencing, particularly at that time, and still today we have to figure out, is not all of those are real. Some of those are errors, and then you have to figure out what's technical errors and noise versus what's real. So out of the hundreds and hundreds of polymorphisms, only 4 were found. So most of them were just mistakes, technical errors that had to be weeded out. And there are ways to do that so you can get there. That's why it takes months to do this work. And those tests were developed. I won't go into the details tests, but each one of those variants had a different morphology colony, and those were identified, and they were sub-components of the total sample, maybe about 0.5% or 1% of the total of the sample. And so these were tested, and I won't go into details, but this kind of network was developed to determine who had those variants in their lab all in one sample and who didn't. And every one of them traces to one source that came out of USAMRIID, which was a flask of culture was being used to develop for lots of different tests. That flask had all 4 of the variants and no other sample does. So that kind of points it to that sample. Now that flask may not be where someone took it from the flask and just went and used it. They could have taken from that, cultured it, and gone a couple of generations away, so someone else may not be the one who used the sample from that flask, but something indirectly. But it points to at least a link to this flask. The problem with that technology of sequencing something, finding a variant, developing a test for the variant, and then typing all the evidence was the best way to do it back in 2002, 2003, and maybe even to some extent today. But it's not a good strategy in the long run because you're only, again, focusing on the one site. There could be other information in the entire genome that could help better resolve some of the ones who had the same 4 variants but maybe they would be excluded for other reasons or vice versa. So what we decided to do is overcome that problem. Just like I said earlier, we have to design a test for, if we had to do it for anthrax, for plague, for Botox, Clostridium botulinum, and all these, would take a long time. You want a test that's a lot more generic. We don't want a test where we have to know what we're looking for ahead of time, and that's where all this next generation sequencing comes into play. So instead of doing the way it was done then, it took months to do, we now have the technology, as I said, to do it very rapidly and to do a lot at one time. And I don't need to know ahead of time what it is if I design it properly. So about 5 years ago, we did a study to demonstrate that this kind of technology would work very well, where I can just take the samples, throw them in, sequence everything, and figure out what it is afterward. So we took 8 strains, which at the time was very challenging. Today could be trivial. 4 of them being Bacillus anthracis, and 4 of them actually being from plague, Yersinia pestis. And then we prepared the DNA, and we threw them all into one tube. Now that's not what most forensic scientists would think of doing, is taking evidence from multiple things thrown in one tube. But we can do that because we have certain bioinformatic capabilities to be able to put things in, tag them, and then pull them apart. So the tags tell us what's apart. So now we can start pooling things and yet not cross-contaminate. So we put all 8 of them into one, and although you don't have to worry too much about where the arrow is, shows the strain, the A2012 is the AIM strain. That's the one that's been sequenced. We know what it is. We did it by our sequencing, and out of 5 million bases, we only had 1 difference in that, which may or may not be real. But that's pretty good if you think about it. It's a 1 in 5 million error, which is very high up there. And I'll point to the other two. These ones had more differences. They're not the AIM strain, so they should have more differences. But when you look at those trees, the two at the top arrow there, it says China and Slovakia, had about 300 differences from the AIM strain, but they were also equally different from the AIM strain, sharing a lot of that, and they're close together genetically. So the data supports that this sequencing technology would allow us to do something very different. So if we could have done this back in 2002, not only could we have sequenced the original evidence, but we could have sequenced the 1,000 samples that we had to go through for less money than it cost us to sequence the original evidence, which was $250,000 a shot, and the few samples that we did. So $1 million plus could have all done all the analysis, all the screening, and in a relatively short time. So technology has changed from our ability to investigate in a short time frame since that attack. So now it happens. Now I mentioned the bioinformatics. Now, if I know most of you, nobody here likes math or statistics, but I think you really need to learn some math, and you really should start thinking about bioinformatics, some computational education, because it is a field unto itself, and unlike some scientists want to believe, this is a science. And there's a lot more science being done bioinformatically, because you can do a lot of simulation studies and a lot of things that define what's necessary. And it's a really important part of the challenge. So let me give you an idea of how this sequencing works to understand why bioinformatics is so important. As I told you, you got 6 billion nucleotides in your genome, 3 billion of each set of chromosomes that you have. So if I put your DNA sequence onto a page, so I made a page, there's 5,000 bases on that page. How many pages do you think it is to make your book you? Any idea? The answer, say a lot. It says a lot of pages, right? Yes, a lot of pages. So if you did it, and you wanted to do the human genome, and you wanted to do it enough times to have confidence that you're getting the right answer, so what we call coverage. When we sequence something once, it has 1x coverage, sequence it twice, 2x. So 6x was shot for in the Human Genome Project just to get to some level. You would need 3 pallets, 40 boxes with 5,000 pages per box to get the 3 billion. And if you wanted to have it to that 6x coverage, you would have to have 18 pallets. That's a lot of information. Now what happens in sequencing? You take that information in whatever order it's in, and you put it through a shredder. You shred it into little pieces, and they're all jumbled up, and you throw it into the machine, and it sequences those tiny little pieces, and somehow it's got to put it back together and figure out what your 3 billion bases are in order. That's not an easy task. Take A Tale of Two Cities, grind it up, shred it. Can you put all those pages back together if you never read the book before? Or even if you read the book before, right? So that's why bioinformatics becomes so important, that if we couldn't do that, we couldn't deal with this. And now what's happened is that we generate so much information that we can't handle the data. And we have to come up with better ways to do that. So we literally are drowning in information, but we're still trying to starve for knowledge. And so as we move forward, this is going to become a significant component of your science. And so from the forensic point of view, this will become critical. Now, to kind of bring it back again, let's talk about cases and talk about sequencing and real-world events. So I asked, do you think that HIV could be used as a bioweapon for biowarfare from bioterrorists? Anybody? Yeah? How many don't think so? Good, one or two. Well, yeah, it could be used, but you'd have to be a very patient terrorist, because if you infect people, many years later something might happen. But it could be a reasonable biocrime, right? And in fact, in a number of countries around the world, it is illegal to have sex with a partner and not inform them that you're HIV positive. It's a criminal act. Threatening people with tainted blood, that's HIV positive, needles, whatever, can be. And if you're in healthcare and you're providing service, could be like a dentist has HIV and he's getting your mouth and such, and you don't do that, those are all criminal acts, and they're punishable for such acts. So there was a case, to give a good example of how microbial forensics will work, Louisiana versus Schmidt, and this was a gastroenterologist who had HIV patients and that, and he was having this this on and off again affair with his nurse, and I guess at one point he decided that he wanted to end it, and allegedly he injected her with HIV and hepatitis C positive blood, and then she came down with HIV and went on from there. So how do you deal with this? Okay, so there's an allegation, there's a crime. Where did the sample come from? They had to start an investigation. So first thing you have to do is you have to ask what is, now remember, he injected her with this. What kind of shot did he tell you she was going to give him? B12, right, there you go. Exactly the same thing. That's what I told you, stay away from B12 shots. Okay, so you have to start asking people in the investigation about their relationships, their contacts. So when you ask a victim, how many sexual partners did you have? She's going to tell you, or maybe not. Depends on what it is. So you're going to have to deal with the limited information that may come out of that. She identified 7 men, including the doctor, and all of them, now you have to go to them and say, well, you want to be tested for HIV. Some may comply, some may not. Well, all complied and all were negative. So that eliminated them as being the source of the HIV. Well, it turns out she's a nurse, and during normal work, she can have hazards. She had no documented needle sticks. There was some splashing of saliva. I don't understand what happened. Maybe someone sneezed or coughed or whatever on her, but she tested negative, and she was a blood donor. So she'd been tested repeatedly up until the point of this B12 injection, and then became positive in that. So when she tested positive, they went to look for the doctor who had a logbook. And guess where the logbook was? It went missing by accident, convenience, whatever. They eventually found the logbook, and one of the last entries was a sample from a patient, and his nurse that works with the samples thought it was strange because the doctor doesn't take the blood and mail it off for testing. He said, I'll take care of this one. So he took the blood and so forth. So now, potential source that may have been the ones to infect the victim. So this isn't a typical sort of, it came from me to her, it came from a tube of blood from a patient somewhere else over to her and that. So now you have to determine what sources are. And to do that, you have to collect samples. And the samples you collect obviously are from the patient, who was the blood source, the victim, and then people in the area who are HIV positive to serve as controls, to see how closely they may be related, to determine whether or not this is a path that could be eliminated from coming from them and be more likely there. So you type them out, and this is just a tree built from sequencing the DNA. And I might say, typical forensic environment, sounds like reasonable, good science, good genetics, but when you go to court, there will always be criticisms. And in fact, some of the defense's criticisms was that not enough was collected. You didn't do enough sequencing. You should have done more. A typical sort of attack that happens even with current analyses in human identification. But the evidence supports, where the little boxes are inside the box, to match to that of the patient being more similar to the nurse victim than any other samples in there. And that was used as evidence in there. He's found guilty in an appeal. The appeal was rejected. And so you can see how microbial forensics is being used in routine work. In HIV cases and hepatitis C cases, probably the most prevalent of these have been used over the last decade in that area. I'll mention quickly agroterrorism, because I keep hitting on food and free stuff and all that you get in the break rooms, but it's not about killing animals. So you understand, it's about disruption, it's about fear, it's about destroying livelihoods of people, economy of nations. That's what terrorism is about. Terrorists don't hate cows. Cows are cute or something like that. They don't want to kill them. They want to do that, but they do want to attack whatever it may be. But it's all about disruption, and it's about causing fear and bringing people to their knees other ways. Agriculture is a little different than it is for humans, because when humans get sick, they complain. they go to the hospital, they go to pharmacies, you see surges, so you know things can happen. When animals get sick, they don't complain, they don't go somewhere. And then you have to have the farmer recognize it and also report it. Farmers don't necessarily always want to report things because it'll affect the sale of their products. It could be the crops could be attacked, could be your water supply, could be in the grocery stores. How many of you walk by, pick up the fruit, look at it, put it back down? Very easy, you know, that. So we have a lot of vulnerabilities. Could be the silos where you store the grain to feed the animals. It could be food handlers in the restaurant, and so on, and the ultimate target could be in your own home in that. So we have a lot of areas we do not have the kind of security to make that a low-risk environment. The reason is if we put the kind of security on there that was necessary to make it so low, food prices would be so high you couldn't afford to eat. So again, there's a balance here, and also the fact that most people aren't terrorists and want to do things. But biowarfare, for attacking agriculture, or crops, or helping crops, is not new. In fact, we practice biowarfare all the time. One of the Bacillus anthracis I mentioned, but Bacillus thuringiensis is a lethal bacteria that attacks insects. And this has been known for a century, and it's used as an insecticide. You buy it in bags, you put it out on your soil, it's all over the place. You probably have all been exposed to it. It's harmless to you, but it's biowarfare, safe for humans and animals, but not for that. Examples have been, as I mentioned, the muffins would be a food source, and that's the nurse that was actually doing that. So if that's your next girlfriend, think twice. No. Yeah, but you have roommates that poison food. We talked about the salmonella in the food. There was a big incident with Chile and some grapes that came in an import that had cyanide on them, and the U.S. government shut down the importing of grapes from Chile. Chile sued the U.S. government and actually won, and it's a very interesting story. So it's not a simple thing of saying we have a risk or whatever. Ricin comes from the castor bean plant, one of easily accessible toxin. In fact, it was the U.S. Army that patented it, so it's all publicly known. So anybody who wants to make ricin can go onto the web and figure out how to do that. So it's used a lot in particularly extortion cases and food tampering. And governments have used bioweapons to attack plants that they consider to be at risk for us, such as the coca and poppy plants. So you can use microherbicides, fungal pathogens that are specific to them. They have low risk to humans, obviously, and to other plants because they're very targeted, but they can help control drug plants and have been used in that. So it's not a new thing. Animal identification is also important. And I got called about, I think it was maybe 4 days before Christmas in 2003, about an incident. Remember BSE in the cattle? You know, mad cow's disease and all? Well, the first incident was a cow in the U.S. was identified, and it was a Canadian cow, not an American cow, so you got to watch those other Commonwealth people that are perpetrating dastardly deeds on the U.S. soil, and that created a big problem, and it kept going and going and going, and whose cow was it? Was it a Canadian cow or was it an American cow? So this created a big problem between the U.S. and Canada on who's to blame. Because if it's mad cow disease, exports of beef is going to stop. So you wanted to figure it was. So as I said, during Christmas 2003, I got called from USDA at home. I was on my holiday time, and they said, we got a problem. How do we prove that's the cow? They wanted to start doing paternity testing on cows. So the traditional thing to determine the source of that. You think it's funny, but actually had genetic markers, set them up, and determined that the evidence supported that the cow was Canadian in origin and not from the U.S. So we won that battle and everybody gave it up. So you can see the future of what's going to happen. We're going to have laboratories that are going to take in samples, whether it's cow blood to soil to food to background materials, and it's going to send it in and develop them and through whether they do one kind of analysis to identify the strain or the species or components of that, and we'll have a whole system of that. Now, to do that, we're going to have to come up with more sophisticated ways to handle all the samples. And we have to do the one thing that forensic science is all about, and that's interpreting data, okay? We get results, and we say, here's my result. But you have to tell people what it means. And if you don't tell what it means, the result is it's just a foreign language to the person. So it's very important to figure out what is there and what you think it means versus what it is. And a lot of times what we think it is not necessarily what it'll be. So I'm always reminded of a missing data story, and I don't know if any of you know this, but during World War II, when the Allies were bombing Germany, they would send their planes over, and planes were being shot at, so when they came back, they wanted to see where they could protect them from the bullets that were hitting them. So when the planes came back, they looked at the planes for where the holes were, and they saw on the body there were holes, so they reinforced to protect the planes. Pretty good idea, right? Yeah, seems pretty reasonable, except for one thing. It has to do with missing data. It's not about the planes that came back that you worry about. It's the ones that didn't come back. And if you might imagine, if it was, if you look at a plane and you got the body, holes in that doesn't seem to shoot down the plane. Holes in the wings and tails. So they were reinforcing parts of the plane that had no value because they were missing data. And that's what we deal with in forensic science. We're always having missing data, but we only focus on the answer being what we see. And that's a big thing we have to think about in there. And biology, there's only one absolute in biology. You know what that is? That there are no absolutes. Biology is mutable, it's constantly changing. Bacteria, evolving all the time. So what you start with isn't exactly the same. So we're never going to get that easy, everything looks the same and hope it works out that we want. So we have to deal with that. In a cubic centimeter of soil is over 10,000 different species of bacteria. So we haven't even begun to determine what are the different types of bacteria that exist in the world and how much variation there is. And in fact, we only know about the oddballs of the bacterial world, the ones we can culture, the ones that infect us. And that's a subset. Most of the ones are not culturable. So we have to develop the methods to identify those, and that's why the sequencing becomes really important. But we have to deal with not knowing everything, but making inferences on source, whatever, and doing something that most forensic scientists don't like to do, say, well, I'm not absolutely 100% on this. There is some uncertainty involved in this. There is some risk, and you have to understand what that is. So we have a data problem we're going to have to deal with. And, pieces of puzzles are going to have to be used to make decisions on very important events. Okay? Now, as I went back to, all that information has somehow to be synthesized into integrating everything into the other evidence in that. So how do you tie together genetic evidence with a teletype that somebody over a telephone said that they're going to bomb a building or something like that? How do you put that into one value so that a decision maker can make a serious, informed decision, which could be attacking another country, invading a country, arresting people, and so forth. So it's a very, very challenging thing. And I always like to make predictions, but today I'm not going to make a prediction because I just reminded of this. Here was a prediction by the Rand Corporation in 1954 of what the home computer would look like 50 years later. They were pretty close, except for a few things. Now, in all fairness, of course, you obviously didn't have a touchpad then or anything, but they did say it would require some unknown invented thing like the computer chip that wasn't around at that time. But you can see is we can make some predictions of what it is, and we're going to move forward with those. But something's going to come up that's going to change it to make it even more practical for us to analyze evidence of some unknown thing in the next handful of years. PCR was an invention that changed the way we look at molecular biology. But if this was what my home computer would look like today, imagine what your phones would be like if you had them now. So with that, I'd like to just say is it's a really challenging field. There's a lot of things to do. It's made great strides, but you can see an idea of what can be done, what still needs to be done in that framework, but you don't have the option of waiting. You have to do it now because an event can occur. But it's all about excluding, associating, and ultimately attributing to is the source or perpetrator of the crime. So with that, I'd really like to thank everybody. Of course, Sam Houston State University and Dr. George Beto and the other Beto, who's not here at the moment, I guess, and then other collaborators who have helped with our projects and that, and then my research team, they're there. So if you have any questions, glad to take it. Thank you.